What is an host cell protein immunoassay, and what is it used to monitor?
A host cell protein immunoassay is a specialized assay that is against all of the proteins of the cell line used to manufacture the therapeutic biopharmaceutical. And so it's a very complicated assay where there's an antibody reagent generated against all of the proteins, or as many of the proteins as can be generated against, and it's used to monitor in process consistency in the purification of that molecule. So what will happen is during the bioprocessing at multiple different steps, samples will be taken, and then this anti-host cell protein immunoassay will be used to measure the level of total HCPs present, and it is used to set the specifications for that bioprocess.
What kind of reporter systems support HCP assays? What reporter systems are most often used?
Yeah, so HCP assays are not limited by reporter systems, and they're frequently, most of the common immunoassay systems are used. So it can start with the basics, which can be either a directly conjugated anti-host cell protein reporter with an enzyme such as horseradish peroxidase or alkaline phosphatase. Could also be conjugated to biotin, and then using a streptavidin HRP or alk-phos reporter molecule. So those are the common basic assays that are used. It can also be engineered to support some of the higher throughput platform systems, whereby the specific reporters for those platforms can just as easily be conjugated to the host cell protein antibodies, and a high throughput system can very easily be engineered from that.
What are some of the critical performance parameters that need to be investigated for an HCP assay?
So some of the critical parameters for development of an HCP assay are common to other assays. In this case, the matrix effects of the different buffer systems is important. This assay is going to see samples that come from very far upstream in the bioprocessing workflow all the way to final product. And during its duty in that workflow, it's going to see samples that are going to be at very acidic pHs. It will see samples that may be at very high buffers, and it's going to see samples which have final formulations and the components of final formulation. So the assay must be able to perform across a quite diverse set of matrices that the sample will be containing. Another key feature of this particular type of assay for host cell proteins is the dilutional linearity. Because the nature or the complexity of the samples with so many proteins with a very wide range, dynamic range of concentrations of those proteins, it can be challenging to get an assay that functions with good dilutional linearity across all of the different steps in the bioprocessing workflow. So that's something that really has to be robustly worked out in order to have a well-functioning assay. So what is the right assay calibration standard? The assay calibration standard should be the same one typically that's used to generate the HCP antibody. So typically what ought to be done is a very large amount of that material should be created as a gold standard material, and then aliquots of that can be used to generate the HCP antibody, and then the other fraction can be put in reserve, and that should be used as the calibration standard for the HCP assay by which the host cell protein levels are determined.
Are there any other recommendations to produce a good reagent and assay?
Are there good recommendations for how to produce both a good reagent and an assay? The way to do that is to begin with the end in mind. Start with thinking about the manufacturing platform that your drug product will be produced from. In the case of therapeutic monoclonals, that's typically a CHO cell line. Consider whether or not you'll have a single program, or if you have multiple different programs, or a platform-based assay by which you need to consider how much material you're going to need to support your products for more than a decade. So those are kind of the critical parameters in thinking about what your end goal is. Backing away from that, you have to start looking at what are the specifics of your platform regarding how your cell line is being used, how it's being grown, where you're going to take your upstream sample from that's representative of your process. If you consider all those things and make your best informed choices about that, it's very likely that you will be able to produce a good reagent in sufficient quantity such that it can produce a robust HCP immunoassay to support all of your programs for the timeline of the life of those projects.